In your analysis journey, some people may think that it ends after assembly and annotation because you have your annotated genome. But really, that's the beginning. And one of the first things you want to do is find out who's close to your genome. You sequence this genome for a particular reason. You wrote the grant, you got the funding, you saw something interesting in it, and you decided to sequence it. And so you want to compare it and try to find what matches that phenotype you saw, what makes this genome interesting compared to other ones. Now, when you look at the BV-BRC resource, there are more than 1.1 million bacterial genomes. So how are you going to find who's close to your genome? Well, there's a special service for that called Similar Genome Finder, and it uses min-hash and mash to find that data. Now, you may be thinking, min-hash and mash, what is that? Sounds like something you would do to potatoes. Maybe, but you can also do it to bioinformatic data. So let's do a quick review of what those are. Let's start with min-hash. So min-hash is the workhorse of the thing, and it's converting sequences into sets of k-mers. And I have an example here of 7 sequences, and let's say you want the k, which is a number. It can be 25, or in this case, it can be 3. You want it to equal 3, and it's going to cut that sequence into all the possible 3s. Then it will hash each of those k-mers, and it's going to assign a number to each of the k-mers. So this hash function is a mathematical algorithm that's taking the DNA sequence, and it's producing a fixed size number for that. And the hash is the output of this. So it's doing it for, in this case, all the genomes in BV-BRC. And you're selecting the smallest hash numbers for that particular genome. So instead of storing all the k-mers, which you can imagine, this would be a huge number. It would take a lot of computational resources to store something that big. It's taking the smallest hash values of that, and this is called the sketch. And this is the min-hash signature. So what it is it's a good random sample of k-mers. Now you compare those signatures between sequences. And here they use something called Checkared similarity, which is looking at the number of shared hashes over the total hashes in the sketch. So that's min-hash. Then we go on to mash. Mash is just taking min-hash and not using alignment, but it reduces all of these sequences into a compressed sketch representation. It's thousands of times smaller than the actual data. So you can imagine that this can run a lot quicker. Could you blast against all the genomes in BV-BRC? Actually, you can't. It's too big. It would take too long, take too much of our resources. You wouldn't be allowed to do that. You could do it at smaller taxa levels, but not across all bacteria. So the sketches are a few hundred values that can be used to simulate a whole large data set of those 1.1 plus million genomes. So what this does is it enables you to cluster a bunch of whole genomes and to rapidly identify, look for similarities, and compare data across those. So you can bring your private data in and look at the public data and see who's closest. So MASH also gives you a p-value, so you know how significant the data is. MASH is computing what's called a minimizer hash, and that's the value of a selected k-mer representing a local region of DNA or protein. In our case here, it's DNA because that's what we're looking at, and it's summarizing those sequences, and then it's estimating a probability that with your private data matching this public data, what's the probability that it's random chance or that they actually match? Now, most of us have taken statistics and we understand a p-value, and if it's less than 0.01, that means it's statistically significant. And if it's greater than 0.05, it means that similarity is probably not meaningful. MASH also gives you a genomic distance between the two sequences, whatever data is you're comparing to whatever genome it says is closest to. So it's looking at really a mutation-based distance between the sequences. And so for MASH distance, if the distance is 0, the interpretation is that these are really close, maybe even the same genome. If it's 0.01, we're talking within the same species. If it's 0.05, maybe the same genus. And if it's greater than 0.1, we're on beyond genera and at higher levels. Now, keep in mind that these are k-mers. It's not the whole sequence. So even if you have something that's 0 distance, because it's looking at small areas of the genome, you can't just say, oh, these are identical, it's the same genome. They're close, but there may be a lot of differences that we're not seeing. So you need to look a little deeper. So this is what the service looks like in BV-BRC. It will take reads, it will take contigs, and it will also take the whole genome, which really is the whole genome. And you can compare it to the bacteria genomes, the viral genomes, and then you can subset whether you want to see the reference or representative genomes, which is a smaller set, or across all the public genomes. And when it returns results, this is the kind of table it gives you. It's mapping metadata, like the collection year, isolation. It's bringing that along for these are the public genomes that the private data was mapped to. And it's showing you the distance and the p-value and also the k-mer counts. It's saying out of the 1,000 k-mers assigned to this particular genome, this private genome matched 665. So let's see this tool in action. And it's one of the few tools in this resource that returns things on the fly, meaning you submit it, you wait and stare at the screen, and it comes up. It doesn't go to a job. It's right there in front of you. So let's get started.